Hello everyone, my name is Jessica Hoffman. I'm a first year in Dr. Jared Call's lab, and today I'm going to be talking to you about does exercise affect brain bioenergetics? First, I think it's important to establish what exactly are brain bioenergetics. Bioenergetics is the branch of biochemistry that focuses on how cells transform energy. This is through the production, storage, and consumption of adenosine triphosphate, otherwise known as ATP. A couple of important measures I'm going to be talking about later are respiration. You'll see that displayed as JO2. This is the flux of oxygen and basically is the chemical process by which energy is harnessed from organic compounds. I'll also be talking about mitochondrial membrane potential, and this is a representation of the proton motive force that drives a generation of ATP from ATP synthase. First, I wanted to establish whether there were substrate preference differences, that is, fuel preference differences among brain regions. I looked at three brain regions, the frontal cortex, hippocampus, and hypothalamus. On our y-axis over here, you can see that flux we were talking about earlier, the respiration, JO2. Basically, this is oxygen consumption and tells us how much energy is being produced by the mitochondria in each brain region. So I first added in ADP, and this was to put an energetic demand on each of the brain regions. I then added a carbohydrate-based fuel that feeds mostly into complex 1, pyruvate and malate. And as you can see, there's a pretty big jump in flux between this ADP addition and the pyruvate malate addition. I then added UK5099, which is a pyruvate shuttle inhibitor. So essentially, this will make it so that any subsequent fluxes I see down the line, I know they're coming from the addition of the next substrate and not from the pyruvate that I've added in earlier. I then added in a fat fuel, palmitoyl carnitine, which is a long-chain fatty acid. As you can see, it doesn't seem the brain really likes to use this as a fuel in any of the brain regions, and it seems to actually flux dipped a bit from that UK5099. I then added in rotenone, which is a complex 1 inhibitor, and this means that only complex 2 thereon would be contributing to the flux that we're seeing. I then added in succinate, which is another carbohydrate-based fuel that feeds into complex 2 of the electron transport chain of the mitochondria. And you can see there was another rise in flux with succinate. And finally, I added an antimycin A, which is a complex 3 inhibitor, and you can see that really squashed any flux that we were seeing. So from this, we can conclude that the brain likes carbohydrate-based fuels, pyruvate malate, and succinate, and I'll be focusing on those from here on. So to bring in the exercise portion of my experiment, I gave 8 animals a running wheel in their home cage for about 3 months, and this was a voluntary running wheel so they could run as little or as much as they wanted. I then followed that up with a treadmill exhaustion test, and in this test, the mice are prodded to keep running until they're unwilling to run anymore, and this would help me to verify whether there was a whole body exercise adaptation. I then followed up with some tissue assays. So to begin, we can see over here, this is the weekly average revolutions of the running wheel of each mouse for about the first 10 weeks. So you can see here, almost all the mice had a really big jump in how much they were running. In the first few weeks, this is a normal behavior that we see in rodents given a running wheel. And then it plateaus around week 5. And then for this graph, this is the graph of my treadmill exhaustion test. And as you can see, my trained animals were able to run about 3 times longer than the sedentary animals until they were exhausted, showing that there was indeed a whole body exercise adaptation. I next wanted to investigate whether there were substrate-specific changes in respiration between my trained and sedentary animals in any of the brain regions. I started out with the hypothalamus first, and I did want to mention that I was limited in the amount of tissue I had for the hypothalamus, and therefore was only able to look at pyruvate. So the way I was able to evaluate substrate-specific changes in respiration was using a CK clamp. And in a CK clamp, you are clamping the brain regions at a specific energy demand. So I started with my first clamp at a higher energy demand, then moved on to a medium energy demand, and finally had a low energetic demand. And unsurprisingly, at a high energetic demand, there is a higher respiration, that is more energy being produced than at the low energetic demand. You can see that my sedentary and trained animals are separated, but this is only my first cohort of animals, and I don't have the numbers to necessarily parse a statistically significant difference between these groups yet. But we'll see what happens with my second cohort that is being trained now. Moving on, I next looked at the frontal cortex to see if there are any substrate-specific changes in respiration. And just as we saw in the hypothalamus, there seems to be a little bit of separation, but not statistically significant yet with pyruvate. And similarly, there wasn't a statistic difference with succinate. Finally, I looked at the hippocampus, and as with the frontal cortex and the hypothalamus, there wasn't a statistically different respiration between my sedentary and trained animals with pyruvate, nor with succinate. I next wanted to see if exercise was going to have an effect on membrane potential in any of my brain regions in a substrate-specific way. So I started with the hypothalamus again, and I was still limited in the amount of tissue I had, so was only able to evaluate pyruvate. And what we're looking at here on our graph is that with the x-axis, we have our membrane potential. And the important thing to know is that as we go towards the left, we're becoming less polarized, and as we go towards the right, we're becoming more polarized. And then our y-axis is that oxygen consumption rate still. And what we see with this is that with the pyruvate fuel, our trained animals are less polarized in their membrane as compared to the sedentary animals. In the frontal cortex, we see, as we saw with the hypothalamus, that the membrane is still depolarized with the pyruvate fuel, and this has now been extended to succinate, as we can see that the trained animals have a less polarized membrane as compared to the sedentary animals. We do start to see some differences in the hippocampus, however, from the frontal cortex and hypothalamus, in that the pyruvate seems to be overlapping a lot more, and with succinate, it's inverted. We see that actually our trained animals have a more hyperpolarized membrane. This brain region had the most tissue samples that had to be excluded from analysis because they did not pass the quality control check. So with my second cohort, we'll see if this reversal is maintained and if this overlap is maintained. So what is the translational relevance of all this? Well, mitochondrial dysfunction is associated with many neurodegenerative diseases, including Parkinson's and Alzheimer's disease. Exercise has been shown to reduce the likelihood of developing these neurodegenerative diseases and also aids in rescue of function in neurodegenerative diseases. The underlying mechanism of this prevention or improvement is poorly understood, however. So understanding physiological changes with exercise may help to elucidate the mechanisms of action in these disease states. Thank you for listening. If you have any questions or are interested in the work that we're doing in the call lab, please feel free to reach out. Have a good day.